found in Japan in 1871. It was first case it was found in Japan in 1871, so it is named as Japanese encephalitis. It is major cause of viral encephalitis. Japanese encephalitis, it is major cause of viral encephalitis. It leads to childhood mortality and morbidity in countries of Southeast Asia and Western Pacific regions mostly. It is reported to cause at least 50,000 cases with 10,000 deaths annually. Cases of Japanese encephalitis globally, it is 50,000, and there are around 10,000 deaths annually due to Japanese encephalitis. Japanese encephalitis, it causes irreversible neurogenic damage. Transmission occurs by bite of mosquito called Culex tritanurhynchus. It is vector-borne disease, so enzootic cycle occurs in mosquitoes such as Culex tritanurhynchus. Reservoirs are ardid birds. Ardid birds means having long legs. You can see this one. So, ardid birds are reservoirs. Amplifying hosts are pigs, bats, possibly reptiles and amphibians. Incidental hosts are horses, humans, and others. Disseminating host, movement of viremic waterboard and movement of humans, it leads to dissemination, and human is dead in the host. Transmission, if we observe transmission cycle of Japanese encephalitis, first pigs or ardid birds. From Culex tritanurhynchus mosquito, it bites to them, and this Japanese encephalitis virus gets transmitted to mosquito. Then mosquito again bites ardid birds or pigs. From them again it reaches mosquito. From them again, in this way cycle will be continued. In case of human beings, if mosquito contains Japanese encephalitis virus after biting ardid birds or pigs, it leads to transmission of human after biting healthy human. And in human, it will be dead in this way. Transmission cycle of Japanese encephalitis virus, it is maintained through mosquito and ardid birds or pigs. Spreading of Japanese encephalitis, it is commonly seen in summer in temperate zone. Countries such as Nepal, Korea, Japan, northern India, in these regions, summer season is the most important season in which there will be spread of Japanese encephalitis. In tropical area, rainy season, there will be spread of Japanese encephalitis. In our concept, in summer season, Japanese encephalitis will be spread most common. There are many countries with epidemic or endemic of Japanese encephalitis. There are various countries, among which Nepal is one. Especially Tarai region of Nepal is endemic. Okay, especially Japanese encephalitis is seen in Tarai region where Culex tritanurhynchus mosquito lives. These are other various countries in which there is endemicity or there may be epidemic of Japanese encephalitis. It is endemic in temperate and tropical regions of Asia. Nowadays, in Japan, there is a reduced prevalence seen in case of Japanese encephalitis. First outbreak of Japanese encephalitis in Nepal, it was seen in 1978. 1978. Almost more than 40 years before, first outbreak of Japanese encephalitis was seen in Nepal. If we observe morphology of Japanese encephalitis virus, it appears like this. You can see in the figure. It is member of Flaviviridae family. Flaviviridae is the family. It is included in group Flavivirus. If we observe morphology of Japanese encephalitis virus, it has three morphologically distinct structure. Innermost core, which is surrounded by envelope. It is enveloped virus. In envelope, it contains surface. It has morphologically distinct structure. It is RNA virus. RNA lies in inner core, which is surrounded by envelope because it is envelope virus. In envelope, it contains surface projections. This is morphology of RNA virus, Japanese encephalitis virus. Already I said it is RNA virus and RNA is single stranded. Diameter of Japanese encephalitis virus, it is 40 nanometer. Diameter of Japanese encephalitis virus, it is 40 nanometer. It replicates in host cytoplasm. Okay, it replicates in host cytoplasm. Morphology is not that defined, and the name flavus is, it is Latin word. Flavus means yellow, which refers to yellow fever virus. It is structurally related to St. Louis encephalitis virus. The outer envelope protein is the protective antigen. In morphology, already we said it is enveloped virus. The outer envelope protein is the protective antigen. It helps in entry of virus to the inside of the host cell. The genome, it is RNA virus. The genome means RNA, which encodes several non-structural proteins also, among which NS1 to NS5, NS1, NS2A, NS2B, NS3, NS4A, NS4B, and NS5. Non-structural, non-structural proteins. The genome also includes several non-structural proteins such as NS1, NS2, NS3, NS4, NS5, etc. NS1 is produced as secretory from NS3. It is multifunctional. It helps in multifunctional protein processing. And NS5, it acts as viral polymerase. Double staining with uranyl acetate and phosphotungstic acid stains the structure of virus well. So we can perform uranyl acetate and phosphotungstic acid staining for demonstration of its structure well. This is electron microscopic view of Japanese encephalitis virus. You can see this picture. So it is electron microscopic view of Japanese encephalitis virus. Now, pathophysiology, how Japanese encephalitis is transmitted from one person to another. First, there should be mosquito bite. Which mosquito? Culex tritaniorhynchus, not other mosquito. Culex tritaniorhynchus mosquito, it should bite, and there should be intake of Japanese encephalitis virus within the mosquito. And if it bites to healthy human, in regional lymph nodes, then there will be primary viremia. Primary viremia, presence of virus in blood first time. Then there will be inflammatory changes in heart, lungs, liver, and reticuloendothelial system. Most infections are cleaved before the virus can invade CNS. Before reaching CNS, most infections are cleaved. When neurological invasion occurs, neurological invasion by growth of virus across vascular endothelial cells occurs. There will be neurological invasion by growth of virus across vascular endothelial cells, and there will be involvement of large area of brain such as thalamus, basal ganglia, brainstem, and cerebellum, hippocampus, cerebral cortex, large area brain, including these parts, it will lead to features of encephalitis. Clinical signs. Already we said there are around 50,000 cases annually globally, and less than one case per year occurs in the US, especially among military and travelers. Incubation period ranges from 5 to 15 days. Remember, annually around 50,000 cases of Japanese encephalitis are seen globally. Incubation period ranges from 5 to 15 days, 1 to 2 weeks. It is mostly asymptomatic, or mild signs can be observed. Mostly Japanese encephalitis or mild signs can be observed. Children and elderly are at highest risk for severe disease. Highest risk is seen among elderly, and high case fatality rate, around 30% fatality cases are seen among elderly people in case of Japanese encephalitis. Clinical signs, they are severe, and it can be acute encephalitis or severe encephalitis. We may observe neuropsychiatric sequelae, or we may observe abortion of fetus. In case of acute encephalitis, there will be abrupt onset with headache. In case of acute encephalitis, there will be abrupt onset with headache, high fever, vomiting. Remember, meningitis, encephalitis, whatever involvement of CNS, there will be stiff neck and there will be stopper, state near unconsciousness. And it lasts for 1 to 6 days. In case of acute encephalitis, there will be abrupt onset with headache, there will be high fever, vomiting, stiff neck, stupor, and it lasts for 1 to 6 days. There may be severe encephalitis, which is characterized by paralysis, seizures, convulsions, stiff neck, chronic sign, followed by coma and death. Severe encephalitis, it is characterized by paralysis, seizures, convulsions, stiff neck, chronic sign, which is followed by coma and death. Now, it may lead to neuropsychiatric sequelae also. Neuropsychiatric sequelae is seen among 30 to 50% of survivors. Among 30 to 50% of survivors from Japanese encephalitis, we may observe neuropsychiatric sequelae. There may be mental impairment, speech impairment, or abnormal movements. So, neuropsychiatric sequelae includes mental impairment, speech impairment, and abnormal movements. If infection reaches uterus, if infection crosses the placenta, it may lead to abortion of fetus. Now, lab diagnosis. For lab diagnosis of Japanese encephalitis, we can perform virus isolation or viral antigen detection, or detection of viral nucleic acid, or detection of antibody, etc. Virus isolation or virus culture can be done. Serum, plasma, blood, CSF, and tissue can be taken and it can be cultured for virus isolation. For viral antigen detection should be done by taking tissue by immunofluorescence. Immunofluorescence means when antibody coated with fluorescent dye is mixed with antigen, it will lead to fluorescing appearance under fluorescent microscope, that is immunofluorescence. So viral antigen detection can be done by immunofluorescence. most reliable test, gold standard. It is detection of viral nucleic acid, viral genome. It is RNA virus, so we need to perform RT-PCR, reverse transcription PCR. For DNA detection, simply we'll perform PCR. For RNA detection, reverse transcription PCR, RT-PCR. So it is ideal method for diagnosis of Japanese encephalitis. Similarly, other lab diagnostic method includes detection of antibody. Detection of antibody. Japanese encephalitis is specific immunoglobulin EM, IgM, either in CSF or in serum. Ideal is in CSF. We can perform IgM capture ELISA. We can perform ELISA and we can detect IgM, immunoglobulin M. There should be four-fold rise in immunoglobulin G, IgG antibody in serum in acute and convalescent phase, which should be done in the 14 days interval. So, detection of IgM in CSF is more important. Sample collection. We can collect blood sample or CSF sample before patient admission, and second sample should be collected 10 days after admission. CSF sample is comparatively more appropriate since IgM may be seen in serum after vaccination also. Due to vaccination, IgM may appear in serum. That's why serum is not ideal sample, but CSF is appropriate. Then we have to perform test as soon as possible. For transportation of sample, we should maintain cold chain, and we should mention type, volume, timing, etc. If we have to store, we should store at 4 degrees Celsius. If we have to store for 1 to 3 days, it means simply in normal refrigerator. We can store if we have to store for 1 to 3 days only. But if we have to store for longer duration, then we have to store sample at minus 20 degrees Celsius. Then we have to send sample to Japanese encephalitis lab immediately. Collection of CSF should be done in 2 vials. First vial for routine pathological examination and second vial for IgM ELISA. Blood sample, either after collection of blood sample, we can separate serum or we can send clotted blood along with serum if lab facilities are unavailable for IgM ELISA. But ideal is CSF sample, not blood. Now, transportation packaging. Already we said we may send to JE lab. How to send? We have to maintain triple packaging system. In triple packaging system, primary receptacle, first there are 3 layers: primary receptacle, secondary and tertiary receptacle. In primary receptacle, specimen that CSF is placed, it should be leak proof, watertight and labeled. Now secondary layer, it enclose and protect the primary receptacles. Adsorbent materials are kept in secondary layer. Then outer shipping package of transport container, ice pack can be placed between secondary and outer shipping package to maintain cold chain. It means in triple packaging system there are 3 layers. In between outer and second layer, we can keep ice pack for shipping, and adsorbent material should be kept for lab diagnosis of Japanese encephalitis, and IgM should be detected. Other test procedures. We can perform plaque reduction neutralization test, PRNT, plaque reduction neutralization test. Most important method is reverse transcription PCR, RT-PCR. It will detect viral RNA of Japanese encephalitis virus, which is most confirmatory. Other supporting test, we can perform peripheral blood smear examination. We can observe severe neutrophil leukocytosis. Normal or slightly raised sugar and elevated protein in CSF can be seen. These are other supportive parameters for lab diagnosis of Japanese encephalitis. Treatment, there is no specific therapy. There is no specific therapy for Japanese encephalitis, and symptomatic management should be done for treatment of Japanese encephalitis. No specific treatment, only symptomatic management. Now, control measures. We can use vaccine, or we can control Japanese encephalitis by vector control. First, vaccine. Name of vaccine for against Japanese encephalitis is SA-1414-2. This is very important question for the MCQ. Vaccine against Japanese encephalitis, it is SA-1414-2. It is attenuated viral strain. It is grown in Vero cells, and we have to inject 2 doses 1 year apart for prophylaxis against Japanese encephalitis. It is effective in preventing clinical disease. So, SA-1414-2, this vaccine, which should be given 1 year apart. It helps in preventing Japanese encephalitis. Other vaccine is JEVAX. It is prepared from Nakayama of Japan, which is formalin inactivated vaccine. Inactivated mouse-derived JEVAX, it was prepared in US in 1992, and 1 ml, 3 doses should be given subcutaneously on day 0, day 7, and day 30. 1 ml, 3 doses subcutaneously on day 0, day 7, and day 30 for JEVAX. Booster after 2 years should be given, but immunity produced by vaccine is short-lived. So SA-1414-2 is more effective vaccine. Control measures include use of vaccine. This is one method. Other is by vector control. We can perform alternative wetting and drying of rice field. As Culex tritanurhynchus mosquito, it lies in rice field. So to control this vector, Culex tritanurhynchus, we have to we should have alternative wetting and drying of rice field, or intermittent irrigation. This is other method. And other method can be used of insecticides to control vector. These are control measures for preventing Japanese encephalitis. This is about Japanese encephalitis.